Now, biotechnology can be used for molecular diagnosis also. Earlier, diagnosis of diseases, for that they used to collect blood samples or urine samples. The problem with these methods are that only if there is a large number of pathogens present in these samples, then only they can be detected. The pathogens should be more in number. But by the time the pathogens number will increase, means the treatment will not be that much successful. It is better if the disease is diagnosed initially so that treatment can be given faster. The molecular diagnosis method. Now, for example, one of the molecular diagnosis method is by using PCR. PCR technique means polymerase chain reaction. Now, this is a polymerase chain reaction machine. So here a blood sample will be introduced into this machine. So what happens is that inside the blood sample, whatever DNA is present, since this machine works under high temperature and they use the enzymes also which are resistant to high temperature, in the presence of that, the two strands of the DNA will separate. These two strands will separate. After that, they add probes that can begin the replication. So there will be DNA polymerase enzyme like Taq polymerase, that is from the bacteria Thermus aquaticus. They are heat resistant. So these are actually RNA. These are RNA, and the new strands will continue from this, and it will be laid according to the complementary bases present on the parent strands. The machine will also have nucleotides present to build up a new DNA strand. So suppose this is the new DNA strand formed. So like that, replication can be done. Now there are two DNA copies here. There's one DNA, there's another DNA. Like that, the DNA copies can be produced up to 1 billion times, or 1 billion copies can be produced through this machine. So if there is only a small fragment of DNA of the pathogen present in the blood, they can be amplified using this machine, using this technique of polymerase chain reaction. And so the diagnosis will be easier. It is possible in the initial stages itself. So if it is diagnosed at a faster rate at the initial stages itself, HIV to AIDS, that transformation can be delayed. Another method to diagnose a disease is ELISA. Enzyme linked immunosorbent assay. In this method, the blood sample will be collected from the patient in order to find out whether they have any HIV virus present. So for that, they can either check whether the antigens of HIV virus is present, or they can check the antibodies that the patient might have produced if there was an antigen are present. So antigen means any part of the HIV virus. So any part can be a protein coat or any part of the virus is called antigen. Or they can even find whether antibodies are produced in response to HIV antigen. Now suppose this is an HIV antigen. So what they add is they have ready-made antibodies. The virus HIV is present here. The corresponding antibodies, when it is put, they will form bonds. Antibodies will link with these HIV antigens. And to make it visible, they use enzymes. They use some enzymes. And if these enzymes are able to stick with the antibodies, that means antibodies are forming a bond with the antigen which is present here, that is HIV antigen, which means that HIV virus is present in the blood sample. Suppose if there was no HIV over here, so if there was no HIV over here, then these antibodies also will not be sticking to those. Then these enzymes also will not be able to be seen then. They will not form the association, which means the patient is normal and he or she doesn't have HIV. The same method can be used to detect whether antibodies in response to HIV is present in the blood sample or not. So if there was antibody in response to this, then when enzyme has been added, they will link with the antibodies present, which means that the patient's blood, patient's body had been producing antibodies against the matching virus, that is the HIV virus. This shows that the patient is suffering from HIV, and then the treatment has to start. Now there is another method that uses autoradiography machine. This is important to detect cancer. There will be, for normal patients, those who do not have cancer, there will be normal genes. With complementary base pairing. Now suppose the patient is having cancer, means these normal genes will not be present, it will be mutated. So to detect whether the patient is having cancer or not, what they do is they will isolate the blood sample and then the DNA, and then they make it into single strands, and they already have the radioactive probes for normal genes that might be complementary to the base pairs present on the DNA strands. So, for example, here it is adenine, thymine, guanine, cytosine. So, the radioactive probe which is used, they may have complementary base like thymine, adenine, cytosine, guanine. So if they are if the patient is not suffering from cancer, means that these radioactive probes will get attached to the DNA strands. And this can be visualized in X-ray film. Because they are radioactive probes, so they can be visualized in the X-ray film. Now suppose if the genes have become mutated, we use radioactive probes. They will not be able to pair up, which shows that the patient has got mutated genes present. And so the patient is suffering from cancer. So that is how molecular diagnosis is done.